These are the derived from the Greek word chroma means color, soma means body. Normally in males have the 44 plus XY. The female also 44 plus XS, autosomes and allosomes. Okay, these are the chromosome. Chromosomal aberrations, the dispersion in the normal chromosome content of a cell. Dispersional disruptions in the normal chromosomal content of cell. In other words, are the changes in the number and the arrangements of number and or arrangements of the genes in the chromosomes. The types of the chromosomal aberrations is the two types, numerical and structural. Okay, what are the numerical changes? Monosome, trisome, tetrasome, triploidy, and next one polyploidy, tetraploidy, different monoploidy. These are the numerical changes. Numerical means number changes. For example, monosome, nullysome. Okay, monosome 2n plus 1. Okay, 2n plus 1. Monosomic is the 2n minus 1. Nullisomic 2n minus 2. Trisomic 2n plus 1. Tetrasomic 2n plus 2. Different cells. These are the number changes, numerical changes. Okay. Next one, structural changes. Deletion, translocation, insertion, inversion. Okay, these are the chromosomal numerical changes and structural changes. What are the numerical changes? Okay, these are the numerical changes. Monosomy. In this one, chromosome is missing. When the chromosome does not separate properly during the meiosis 1 and meiosis 2, is called is the mitotic non-disjunction. Okay, it is a incompatibility with the survival basis, loss of the much genetic information, monosomic actually numerical change also euploidy and aneuploidy. Euploidy and aneuploidy. Aneuploidy, they can divide the monosomic, nullisomic, trisomic, tetrasomic. In euploidy, these are the monoploidy. Okay. Next one, polyploidy, triploid, the tetraploid, auto. Okay, these are the euploidy and mainly chromosome aberrations are can be broadly the variation structure and the chromosomes, the variation number of chromosomes. Most of the sexually reproducing the eukaryotes diploid with two sets of the chromosome. Euploid variation may extend from the haploid or monoploid condition 1, the various levels of the polyploidy. Numerical two types, euploidy and euploidy and next one aneuploidy. Euploidy means monoploid. The organism with one set of chromosome in the somatic cells are called monoploidy. N. Okay. The is given to n number of n. The chromosomes, organisms with one set of chromosomes in their somatical cells are called monoploidy. These are the characteristically found in the nuclei of some lower organisms such as the fungi. Monoploid condition also they can see into the in some of the fungal members. Monoploid is the higher organisms, is the especially is the rare. Most of these are seen to the lower organisms. We can see to the rare in higher organisms. The notable exemption exists the male bees and wasps. Monoploidy plants are known as the usually sterile condition. Monoploid plants are the most of the sterile condition. Okay. Next one, euploidy, polyploid. Polyploid levels is higher than tetraploid are not the commonly encountered in the natural population. Some triploids as well as the tetraploids exhibit the more robust phenotype. There is the diploid counterparts. Many commercial fruits and the ornamental are the polyploidy. These are the very important. Many commercial fruits And next one is the many commercial fruits and the ornamental plants are the pulpilide condition, pulpilide. Sometimes specialized tissue when the diploid organism will be polyploid. For example, some of the liver cells of human beings are the polyploid. A familiar example, polyploid and endosperm tissue also polyploid. Endosperm is the 3x stage. Okay, the endosperm tissue of the corn and other grains. Polyploids offer the opportunity for the studying of the dosage effect. The two or more alleles are the one locus. We have the present of the single locus, the alternative. Mainly polyploid condition. Mainly they are seeing to the larger leaves and the flowers and fruits. Mainly these are seen to the apparently having the larger leaves, flowers and fruits. Many commercial fruits and the ornamental plants they can see to the polyploid condition. Example, liver cells of human beings are the polyploid condition. Next one, endosperm also the polyploid condition. Endosperm is the 3x stage. Okay, endosperm is the 3x stage. Okay, triploid. What is the triploid? Three sets of the chromosome can originate by the union of monoploid, union of union of monoploid. These are the gamete is N with the diploid gamete. The extra set of chromosomes are triploid distributed in the various combination of the germ cells, resulting in the genetically unbalanced gamete. Genetically unbalanced gametes. Because of the sterile, there the characters is triploid. They are the common in the natural population. Tetraploid, four set of the chromosome can arise in a body cells by the somatic, especially euploid condition also they can see to the in somatic cells. Most of the somatics, monoploid condition only they can see the lower organism. Example is the fungi. Polyploid condition they can see into the some of the fruits and the ornamental plants. And next one is the larger leaves and the flowers and fruits and endosperm also. A triploid condition they can see into the germ cells. Okay. Next one, the tetraploid condition also they can see into the cell to the chromosomes arise body cells of the somatic doubling of the chromosome number. Doubling accomplished either spontaneously or cannot be induced by high frequency export the chemicals such as the alkaloid, colchicine. Tetraploid produces the union of the unreduced diploid 2n gametes. Actually, the colchicine is the colchicine atom you can use for the doubling of the chromosomes. Doubling of the chromosomes. Okay. Next, no, autotetraploid. Allotetraploids is the both are the two types of the auto. It indicates the polyploid involves the only homologous chromosome sets. Chromosome sets. Auto mean especially the indicates the polyploid involves the only homologous chromosome sets. Somatic doubling of the diploid produce four sets of the homologous chromosomes. Union and underweight diploid gametes from the same species to be accomplished the same results of the mitotic chromosome. Next one is the allotetraploid. The organism crossing the different species are the term is the allopolyploid. Polyploids that originate from the crossing between the different species. Our term is the allopolyploid. Allo indicates the non-homologous chromosomes. Auto indicates the homologous chromosomes. Allotetraploid indicates the non-homologous chromosomes. The nano is are mainly is are the involved in the non-homologous chromosomes are involved. Allopolyploid is the individual derived from the intraspecific hybridization from the two or more genetically distinct related diploid species. Double diploids of the kind normal chromosomes. If there is numerical aneuploid, then polyploid. The structure abnormalities, the deletion, duplication, inversion, translocation. Numerical aneuploid, the variation in the number of particular chromosomes within a set, especially variation in particular chromosomes within a set. Hyperpolyploid, gain of the chromosomes. Trisomy, 2n plus 1. Double trisomy, 2n plus 1. Tetrasomy, 2n plus 2. Hyperpolyploidy, monosomy 2n minus 1, double monosomy 2n minus 1, nullisomy 2n minus 2. Okay, polyploid condition, the condition which is the cells have the more than two homologous set of the chromosome. Triploid 3n, tetraploid 4n, pentaploid 5n. Okay, the aneuploid six chromosomes, Klinefelter syndrome also XXY is the one X chromosome is the extra chromosome. Okay, these are the trisomy, the aneuploid, especially somatic cells. We can see these are the occurs into 1 in 5,500 to 1,000 members in the. These are the sterile condition, no obvious facial dystrophy, narrow should be the with hips. Okay, Klinefelter syndrome. Next one is the Klinefelter syndrome also. Smaller testes. Next one is the and penis has decreased the sexual interest. The weaker bones, lower energy. Breast growth also is the very less facial and the body hairs. Okay, reduce the muscle tone. Jacob syndrome is also aneuploid of sex chromosome, aggressive due to the high level of the testosterone. Prone is the violent while beating. Adult male could be the more impulsive and the emotionally mature. The Jacob syndrome is occurs into the 1,000 people only one seen the one people. Lanky, clumsy, anger, dyslexia, develop reading disorder, problem in motor coordination, behavioral problems, behavioral tantrums, more impulsive. Okay, these are the Turner syndrome. Aneuploid the sex chromosome demonstrates the visual defects, including the poor handwriting and likely underdeveloped to the arithmetic skills. Okay, Turner syndrome, monosomy part 7, the short stature, ovarian failure, otis media, early OMT is the dyslexia reading disorders. Potato also syndrome is also tri syndrome. Several intellectuals, the disability, physical, Edwards syndromes. Down syndrome is the trisomy 2 and 21. These are the trisomy, monosomy, trisomy. The next one, tetrasomy 2n plus 2, addition of the two chromosomes, two chromosome pairs at the particular chromosome. A cell contains the 48 chromosomes. The tetrasomy, the condition is the tetrasomy. Tetrasomy also can be due to the egg is fertilized the sperm with the double set of the chromosomes. The egg is fertilized by the two sperms. The egg is producing the cells fail the divide the properly. So in the mature egg contains the double set of the chromosomes. Triploidy, tetraploidy. Okay, these are the next one is the aneuploidy. Aneuploidy is the addition of or subtraction of the individual chromosomes from the normal complete of the somatic chromosomes. Term of the aneuploidy. And the aneuploidy variation involves the single or two sets of the chromosome. Aneuploidy can be divided into four types: monosomics, nullisomics, trisomics, and tetrasomic. What is the meaning of the monosomic? Loss of one whole chromosome in the otherwise the normal chromosome complex in the term as the monosome is indicated to 2n minus 1 loss of one set of chromosomes. Okay. loss one or whole chromosome in a otherwise normal chromosome complaint termed as the monosomy. The single chromosome without pairing the partner may get to the either pole during the meiosis, but more frequently will lag at the anaphase of the pairs. They include the adrenals. Monosomics also two kinds of the gametes. One is the normal and then another one is the n minus one. In plants is the n minus one gametes is the cell length function. Nullisomic, nullisomic also it indicates the 2n minus 2. Loss of one pair homologous chromosome in normal chromosome compliant. The term also nullisomic. Nullisomic can be designated by the 2n minus 2. Loss of pair chromosomes in a lethal diploids. So polyploid, however, it can tolerate loss of two homologous set chromosomes. The still survive the, for example, nullisomics, the exapolyploid with 6n minus 2, especially with in the 6n minus 2 exhibit the reduce the vigor and fertility of the can survive the to maturity. The next one trisomic addition of the one extra chromosome a normal chromosomal trisomic 2n plus 1 is mainly 1 2n plus 1 addition of one extra chromosome. Okay. Mainly is that the n plus 1. If two chromosomes of the trivalent go to the one pole and the third goes into the opposite pole, then gametes will be n plus 1 and the n respectively. Trisomy can produce the different phenotypes depending upon the which chromosome of the complex is present in the triplicate movements. The presence of the one extra chromosome, autosome 21, of the tetrace resulting in the Down syndrome, formalism or Mongolism. Tetrasomic indicate the 2n plus 2, the addition of the one extra pair of homologous chromosomes. The normal chromosome complement, the tetrasomic condition is excess of 2n plus 2. Next one is structural abbreviations, deletions. Okay, first one is the deletion. Deletion, it means loss of a small portion of the chromosome. Loss of a small portion of the chromosome is also called as deletion. It occurs from breakage at the random in both chromatids of the chromosome or only in one chromatid in break. Deletion. These are the deletion. See, this one is the small portion of the chromosome deleted. Okay, the small portion of the chromosome, it means the loss of small portion of chromosome deletion. Okay, next one is the deletion also. The pollen is the applied to the stigmas, the plant carrying the recessive alleles at loci, which may be in the region of the induced deficiency. In the progeny, the few plants will show the recessive phenotype. This phenotype is the indication that recessive allele carried hemizygous condition due to the deletion in the corresponding loci and the chromosome from the irritated pollen. Mainly is that the eternal deficiency results in the unpaired end region. X-ray induced the deficiencies occur either heterochromatin or euchromatin. Next one is the translocation. Next one is the translocation, the resulting of the reciprocal exchange of terminal segment of non-homologous chromosomes. Sometimes a segment chromosome becomes detached and units with the another non-homologous chromosome. Translocation means sometimes a segment of a chromosome becomes detached and unites with the another non-homologous chromosome. These are the two types: simple translocation, reciprocal translocation. These are the translocation. Okay. So before translocation, and next one is the after translocation. So this one the exchanges, especially reversible, reversible changes, reciprocal translocation, and the simple. These are the simple translocation. Mainly, the origin may be natural and induced dependence on the breakage and the reunion of the breakage and reunion of chromosomal fragments. Chromosome breaking may increase the with age. The old seed germinated and grown out provided they have the translocation wheat and the barley seed. Other causes including the interlocking of bivalents, okay, during the zygote in the packet in one, the translocation, reciprocal inversion. What is the meaning of the inversion? Inversion means a fragment is transferred from a chromosome to non-homologous chromosome. Mainly a fragment is the transfer from chromosome to non-homologous chromosome. Chromosome to non. It is the rare and the non-reciprocal type of the translocation, which involves three breaks. Okay, insertion. For example, XY is the insertion, inversion, or insertion. Inversion, it results when there are two breaks in a chromosomes and detached segments because the reinserted in the reverse order. It is a it is of two types of the depending of the inclusion or the absence of the centromere. Paracentric, both breaks one arm chromosomes. Paracentric. The pericentric is the break points on either side of centromere. Paracentric mean both breaks in the one arm of the chromosomes. Next one is the pericentric mean it has the break points on either side of the centromere. Okay, these are the inversions. Next one is the isochromosome. It is the chromosome that has lost one of its the arm and replaced with the exact copy of other arm. Ring chromosome. So the first discovered by the Lillian Wagon. Structure of deletion, amount of genetic information the chromosome can change, duplication, inversion, translocation, deletion. See, this diagram is the loss of the region of the chromosome. A chromosome deficiency occurs when the chromosome breaks and the fragment is lost. These are the single break, the two breaks, deletion, deficiency. The phenotype consequence of the deficiency depends on size of the deletion, functions of the genes deleted. Phenotypic of the deletion usually determine two types of the deletions. Cry chart syndrome. This is high-pitched cry, intellectual disability, delayed the development, small head size, the low birth weight of the weak, muscle on infant, George syndrome, Engelmann syndrome, duplication. What is the meaning of the word? A chromosome duplication is usually caused by the abnormal events during the recombination. Okay, abnormal event of the recombination. Duplication. A small duplication of not a complement combined by the phenotypic effect. Phenotypic effect. Large duplication produces the phenotype through the imbalanced gene dosage and tend to be less eternal constant, the major force of the genome evolution. Inversion, a segment chromosome that is the flipped relatively to the in the homology. So breaks in the one chromosome is the flip condition. Pericentric, paracentric. Okay, paracentric involves the only one arm, involves the P and Q, both arms. Inversion. arise from the chromosome. Entanglements and the breaking during the mitotic phase is also from the recombination between the transposal elements. Translocation, reciprocal translocation. When segment one chromosome becomes attached to another, translocation. Reciprocal translocation. What's some translocation? Isochromes entering chromosome. Mirror image of the chromosome or loss of the arms with the duplication of other. Breaks occur within the both arms of the chromosome. The two broken ends enable to the two eccentric fragments are lost. Results in the double deletion, epilepsy, mental reduction in the chromosomal abnormalities. Duplication. Let we discuss the duplication. Extra chromosome. Simple means part of the chromosome is duplicated, present in two copies. Duplication, extra chromosome. Duplication. Okay, these are the chromosome abnormalities, versions and the chromosomal abnormalities. Okay, next one is the mutations. Okay, what is the meaning of mutation? A mutation is a change in a DNA sequence or nucleotide sequence. Mutation is a DNA change in a DNA sequence or nucleotide sequence. A change occurs in the number or structure or gene or a chromosome referred to the genetic mutation. Whatever it may be, member change or structural change or gene change, any types of the change. Mostly DNA is a nucleic acid present almost every organism. It's made of the long chain of the nucleotides, a polynucleotide chain. The mutation can also change gene expression, change or the mutation in the structure or number of chromosomes, also resulting in the genetic abnormalities. Mutation can result from the DNA copying mistakes made during the cell division, exposure to ionizing radiation, exposure to the chemicals is called mutagens, or infection by viruses. Mutants, and mutagens. The mutation is an important biological process in nature. It can be helpful or harmful, maybe helpful or harmful. The mutation creates the variation in nature by providing new alleles and hence helps in evolution. A sudden or undesirable mutation can cause the cancer and the other harmful genetic disorders and suddenly changes. For example, growing of the cells, abnormal cancer, it leads to the cancer. Okay, sudden undesirable mutation can cause the cancer and the other harmful genetic disorder. Now, mutation means due to the replication of errors, expose the mutagens and the viral infection, change or alternation occur in a DNA sequence which causes the genetic abnormalities is known as the mutations. The mutation, the word was originally derived from the Latin word muture, the meaning to change. The term mutation was coined by the Hugo de Vries in 1890. The mechanism of the mutation was studied by the Morgan in 1910. In 1927, H.J. Muller performed the experiment of the artificial mutagenesis. Artificial mutagenesis introduced the mutation in Drosophila and awarded the Nobel. Hugo de Vries in 1946. Ends of the mutation. The base of the type cells is the somatic mutation, gametotic mutation. On the base of the origin, these are the types of the cells, somatic and gametic. On the base of the origin, spontaneous mutation, induced mutation. On the base of the size and the quality of the mutation, gene mutation, chromosomal mutation. Different types of the kinds of the mutation. On the base of their cells, somatic mutation. What is the meaning of the somatic mutation? This type of the mutation found in the somatic cells or non-reproductive cells of the body is known as somatic mutation. They never inherit. These are the never inherit. Okay. Mainly this type of the mutation occurs in the somatic cells. Somatic non-reproductive body cells. Okay. That the somatic. It is the never inherit gametic mutation. This type of the mutation found in the only in germ cells or reproductive cells. It is also known as gametic mutation. They always inherit one generation to next generation. These are the transform to one generation to another generation. Okay, they always inherit one generation to next generation. And the next one is on the basis of the origin, spontaneous mutation. Spontan this type of mutation is spontaneously produced in the organisms by the effect of the various factors like environmental, like radiation, temperature, pressure, different types of the radiation, for example, UV rays, X-rays, temperature, high temperature, pressure also. The base of the origin, spontaneously, okay, suddenly changes. Suddenly changes in some of the organisms, the various factors, especially radiation, temperature, and the pressure. Induced mutation. In this type of the mutation are artificially produced in the organisms. Some chemicals, X-rays, gamma rays. Okay. The factor inducing mutations are the called mutagen or mutagenic agents. Especially induced mutation, they can apply for the some of the chemicals, gamma rays, X-rays. Okay. This is a highly affected induced mutation. The spontaneously mutation mean especially spontaneous suddenly changes. The induced mutation they can apply the some of the mutagens or the chemicals. And then on the base of the size and the quality mutations are the another two types, gene or point mutations. Gene or point mutation. What is the meaning of the gene and the point mutation? Mutation are series of mutation occur in the polynucleotide sequence of a gene or single base of the DNA that changes the function of it, refer to gene mutation. A point mutation are the base pair of substitutions. Mainly these are the point mutation mean mutation or series of mutation occur in the polynucleotide sequence of gene or single base of the DNA. Mainly changes in the DNA deoxyribonucleotide. Deoxyribonucleotide. The gene mutation or point mutation. Point in the single, especially they can change into the in polynucleotide cells, a DNA molecule. Point mutation also two types. One is the substitution mutation, another one the frame shift mutation. Substitution mutation means when one type of the nucleotide base is substituted by the other type of the nucleotide base. For example, is also called as a substitution mutation. For example, purines. Purines, what are the purines? Adenine and guanine is the both are the purines. And next one is the uracil and thymine. Sorry, adenine and guanine, thymine and cytosine both are the pyrimidines. The mainly substitution mutation mean substitute by the other type of the nucleotide base, especially one nucleotide base. For example, the both are the nucleotide bases. How many is the nitrogen base also nucleotide base especially? Okay, mainly the nucleotide base is substituted by the other type of the nucleotide base is also called as substitution mutation. Frame shift mutation. It is a type of point mutation. Base pair alteration causes abnormality reading frames, which ultimately results in an abnormal protein formation with one or two bases added or deleted. All of the three bases codons changes. There are the gene point mutation. There are the two types of the substitution mutations, transition mutations, and next one is the substitution transition and the transversion mutation. What is the meaning of the transition mutation? It occurs when the pyrimidine base, thymine, and the cytosine, the substitute for the another pyrimidine base, and the purine base, when a purine base, adenine or guanine, substitute the another purine base. Okay. Mainly transition mean it occur the pyrimidine base substitute for the another pyrimidine base, or when purine base substitute another purine base. For example, A replace G, T replace C. Only transition mutation mean replaced by the pyrimidine place to pyrimidine. And next one is the purine place to purine. Okay, this is a transition mutation. And transversion mutations, it occurs in a purine. It occurs when a purine base substitutes for the pyrimidine base or vice versa. Okay, transition mean only they can change the replacement of the pyrimidine to pyrimidine. Okay, and next one transition the purine to purine. The transversion mutation mean it occurs the purine base substitute by the pyrimidine base is vice versa, and pyrimidine base substitute by the purine base. Okay, when TA is are the CJ pair replaced by the AT pairs. Okay, these are the transition mutations. And point mutation, substitution, already we discussed the substitutions. And some of the base of gene sequence are the replaced by the other bases. There are the three types of the substitution mutations: nonsense mutations, missense mutation, and silent mutation. Okay, nonsense mutation, missense mutation, and silent mutation. Substitution mutation. What is the meaning of the nonsense mutation? It occurs when one nucleotide is the substitute and the leads to formation of the stop codon instead of codon that codes for the amino acid. Nonsense mutation. Mainly nonsense mutation, it occurs when one nucleotide is substituted and this leads to the formation of a stop codon instead of a codon that codes for the amino acid. Nonsense mutation. Nonsense, nonsense codons, stop codons. Okay, UAA, UGA, you these are the three nonsense codons is there. Then missense mutation occurs when nucleotide is the substituted and different codon is formed, but this time the codon that forms is not a stop codon. Instead, the codon produce a different amino acid in the sequence of amino acids. Okay. Next one is the silent mutations. What is the meaning of the silent mutation? The nucleotides is the substituted but the same amino acids produced anyway. This can occur because multiple codons can code for the same amino acids. Mainly, we can change the same amino acid. Nucleotide substitute by the same amino acid produced anyway. Duplication, and some bases are base duplicated in the sequence. There are the duplications. Inversion, and see these are the duplications. The number of duplications, and inversion when some gene sequences inverted are inserted back into the original sequences. There are the frame shift mutation also. A frame shift mutation occurs when the entire reading frame is altered due to the single base substitution. Point mutation. A reading frame is the sequence of all the codon between the start codon AUG and the stop codon. AUG is the starting codon, or Age is the starting initiation codon. The point mutation, insertion or deletion in the reading frame changes codon with it in the thus the protein synthesis altered to type the point mutation is also called frame shift mutation. Next, inversion, insertion, addition of the base into the gene sequence, often is addition mutation 22. Deletion, delete the some base pairs, delete the gene sequences. Chromosome mutation and abortion. Mutation that is the occur more or on the gene or the called chromosome mutation because the affect the structure, function, and inheritance of the old DNA molecules. These chromosome mutations results from one or more breaking in the DNA molecules of the genome from the exposed to radiation and the wrong rejoining. Types of the chromosome mutation according to the change in the number, aneuploid, polyploid, we discussed the last class, previous slides, aneuploidy, polyploidy. Section of the mutations. Next one is the molecular base of the gene mutations. Gene mutation, point micro mutation. A point mutation is the single base substitution. It's a type of the mutation that causes the replacement of the single base nucleotide with another nucleotide by the genetic material, DNA or RNA. Often the term point mutation is included the insertions, deletion, and single base pairs, transition, transversions. Next one, frame shift mutation, deletion mutation, insertion, transversion, tautomerism, ionization mutation, base analogous, deamination, alkylation, depurination, especially induced gene mutations also, physical mutagens, X-rays, gamma rays, alpha particles, and next one is the ultraviolet ray, electromagnetic radiation with the wavelength shorter than the visible light along with the X-rays can be induced. Radiation was the first mutagenic agent known effect of genes when the first reported in 1920s. The radiation itself as the discovered in 1990, Brandgen discovered the X-rays in 1895. They sequentially discovered the radioactivity in 1896 and the Marin Pierre discovered the radioactive elements in 1898. Source of the radiation. What are the natural source of the radiation produced so-called background radiation? This include the cosmic rays from the sun and the outer space, radioactive elements, and soil and the terminal products, and in the atmosphere once exposed due to the background of radiation varies with the geographical location and addition humans have created the artificial source of radiation. Among these are the medical testing and the nuclear testing of the plants and various other products taken together the overall. Electromagnetic spectrum, the visible light of the electromagnetic spectrum, especially uh, the spectrum radiation also 10 to the power of 3 to 10 to the minus 5. Mainly the visible lights also they can see to the 10 to the power of 10 to the power of 3 to in the minus 10 to the power of 3 1. Mainly 750 nanometer, 380 nanometer is the visible range. The X-rays also 1 nanometer. The gamma is 10 to the power of minus 3 nanometer. Cosmetic rays 10 to the power of minus 5. Okay. And the radio waves What are the roles of the mutation? Mutation is a very important for evolution. So, sorry, chemical mutations also molecular mechanism. They can affect of the chemical mutations, mainly uh, bromomethyl uh, enol form Bu bromomethyl, the bromodiaxmin BUDR. Next one, two amino purines. Next one, inhibition of the nucleic acid precursors. Chemicals which is the structure of the pairing of the property of the base. Nitrous oxide, these are the chemical mutagens. Mainly nitrous oxides also. Next one is the alkylating agents also they can use for the next one hydroxyl amine, the chemical mutagens. And DNA repairs. These are the two types, mainly DNA repair also, because DNA damage occurs spontaneously and the result is ubiquitous environmental agents. Most of the organisms have some the capacity repair of the DNA. DNA is the only macromolecule in the is prepared by the cells. When divided in the repair mechanism into three categories: damage reversal, removal, and next one is the damage tolerance. What is the meaning of the damage reversal? Simplest enzymatic action restores the normal structure without breaking backbone. Okay, simplest enzymatic action restores normal structure without breaking up the backbone. Damage reversal. Damage removal involves the cutting out and the replacing the damage or in a appropriate base or section of the nucleotides. Damage tolerance is not truly repair, but it is the copying with the damage that can go on. Okay, damage reversal is also called as the and many types are photo reactivation or the direct replacement mainly, and next one the ligation of the single strand breaks, single strand, and next one is the damage removal is the base execution repairs. The mainly the damaged or inappropriate base removed from the sugar linkage is replaced. These are the glycolysis enzyme, but the base the sugar bond. Uracil glycolysis enzyme, which is removes the uracil from DNA, sorry RNA. Uracil is not supposed to be the DNA. It can occur in the RNA primers, not removed in DNA. The replication in cytosine determinate. The enzyme recognizes the uracil and the cuts on the glycosyl enzymes. The sugars then cleaved in the new base of the DNA, polymerase using the other strand of the template. Mutants lacking the uracil glycolysis have the elevated spontaneous mutation levels and are the hypersensitive killing and the mutation by the nitrous, nitrous acid. There are the two other specific glycosides radiation in the chemicals. A mismatch repair, nucleated execution repairs, DNA damage tolerance. Not all the DNA damage can the removed the immediately. Some of the it many persist for the while. If DNA replication fork encounters the DNA damage, such as the pyrimidine dimer, it will be normally block the further replication process. Some of the what are the useful of the mutagens? Some of the mutagens are the chemical or physical agents. It increase the frequency of the mutation. Essentially, all the mutations show the some specific for the type of mutation produced. The following the table spontaneous mutation mechanism, DNA replication and the repair the errors, spontaneous modification of the nucleotides. Type of the mutation produced, all types of the mutation produced in the spontaneous mutation. UV radiation, pyrimidine dimers induce error the prone repairs SOS. The mainly GC or the AT is the transmit, but the all other types of the mutation, including the deletions, frame shifts, and rearrangements, the somewhat lower frequency. 2 amino AP, the mainly is that the mechanism base analog. AT to GC, GC to AT is the transition. Okay. Next one, bromouracil, the base analog. GC to AT, AT to GC is the transitions. Hydroxylamine, NH2OH, alkylating agent is the generated the mechanism. And then is the GC to AT in the transition multiple closely spaced mutation common. And ethylomethane sulfonate, alkylating agent generated the O-methane GC to AT remains. Okay, next one, nitrous acid. Next is the oxidative detonation, Gc2AT and the AT to Gc transition produce the lower. Uh, these are the what are the roles of the mutation? Mutation is very important for the evolution. It provides heredity variation. The variation produced by the mutations accumulated over a long time period and gives the development of new species. Mutation help in the survival of the species in change of the environment. Mutation help the organism and adapt the self according types of the mutation, chromosome gene mutation. What are the importance of the mutation and disadvantage? What are the uses and ownership mutations? The advantage, some of the variety caused by the mutation by the extremely beneficial an organism or species. Mutations frequently result in the production of the proteins with new or altered activities that can be beneficial on the organism. This is the change in the environment. Mutations for the examples help many insects withstand the chemical pesticides. Next one, the microorganisms have been able to adapt a new materials in the environment due to the some mutations. Polyploid plants are the typically larger and the more resilient in the diploid plants. Important crop plants such as the create polyploid plants are typically larger and the more resilient in the diploid plants. Important crop plants such as the bananas, limes have been created in this manner. The polyploid occurs naturally, citrus plants as well as the frequently result in the spontaneous. What are the disadvantages of the mutation? Some of the most harmful mutations are those the alter protein, the structure of gene function drastically. Next one, this mutation produces the faculty proteins which can be impair normal biological activity. These are the results. Hereditary disease. These are the one of the disadvantages of the mutation. Some cancer, for example, are caused the mutation that induce the uncontrolled of the cell growth. These are also one type of the disadvantage of the mutations. Next one, so alteration in the morphology of red blood cells are the characterized sickle cell disease, and red blood cells are the normally spherical. Sickle cells are the long and pointed appearance. Anemia, intensifying recurrent infections is the standard growth of the all indications of the condition. These are the importance and advantage, disadvantage of the mutation. In this class, we will discuss the types of the mutations. Next one is the chromosomal abnormalities. Thank you.